Good afternoon and welcome to the launch webinar for the BBSRC Follow-on Fund. My name is James Phillips and I'm pleased to be speaking to you today at the start of our August 2025 round. The Follow-on Fund, or FOF, is one of BBSRC's flagship programs with a fantastic record of investing in researchers who translate bioscience ideas towards a huge range of products, services and processes to generate both economic and societal impact. Today's webinar will start with an introduction and strategic overview of the fund. We'll then present examples of case studies from successful applicants. After that, we'll describe how to make your application, and if you're joining us live, you'll be able to take part in our online Q&A session. Since 2004, the fund has invested approximately £80 million in more than 500 projects to drive bioscience discovery through to outcomes such as products, services, spin-outs and licensable IP that has benefited the UK both economically and socially. Awards take the form of a UKRI commercialisation grant, and eligible researchers from UK universities and institutes are able to apply for up to £800,000 to advance their idea. The fund makes awards twice each year, with opportunities opening in both the spring and summer. This slide summarises the support available. The follow-on fund will make grant awards that allow standard costs from a total fund of £3 million in this round. Awards may range from £100,000 to £800,000 in total. Please note that this is the 100% FEC figure. We estimate that the earliest start date is the 1st of July 2026 and that awards may run for up to 24 months. This time, the closing date is the 15th of October at 4 p.m. Please do remember to submit your application to your internal approvers before that date. Late applications will not be accepted. The next round of the fund is planned for late January 2026. For those of you who are familiar with previous rounds of the programme, this is a quick update on recent changes. To speed up the time to process the awards, applications will now go directly to panel review without the external peer review stage. The size of the assessment panel has been increased to facilitate this. The requirement for applicants to hold a previous BBSRC award has been expanded to include any direct previous BBSRC funding. Guidance on managing intellectual property and project partner contributions has been updated to be clearer and more flexible. In the last round, we highlighted the UKRI Proof of Concept Fund, which has now closed for assessment. We continue to collaborate with colleagues to ensure a joined-up ecosystem and will continue to explore the connectivity between the two schemes. Finally, we are pleased to currently be piloting a link into the BBSRC iCure programme. Existing FOF award holders have been invited to participate in an iCure Discover cohort to build entrepreneurship and commercialisation skills. We intend to offer opportunities to join future iCure rounds to future FOF award holders, and we would encourage you to consider this when developing your future project plans. Successful applicants may be contacted with further details. You can find more information about these changes on the BBSRC Funding Finder webpage for this round. The fund is managed by a team that I'm pleased to introduce. The fund's strategic portfolio manager is Faye Shannon, and the senior lead for the delivery of the fund is Ellie Tayaki, who supports the fund's portfolio manager, Vicky Jill. We very much welcome your queries. However, please note that the opportunity is delivered through the UKRI funding service. If your question is specifically about how to use TFS, then we would encourage you to please email the TFS support desk directly. For questions that are specific to your project, so for example, if you want to check whether your idea is within the remit of BBSRC, then we would encourage you to send us an email at our team mailbox. We prefer to answer queries like scope, remit, and arrangements for intellectual property by email, so that there is a clear written record of any advice issued. The next few slides provide a strategic overview of the follow-on fund in the context of knowledge exchange and commercialisation, both in UKRI and BBSRC. The fund is an investment in bioinnovation by BBSRC, the Biotechnology and Biological Sciences Research Council. We have a track record of providing dedicated knowledge exchange and commercialization activities to unlock the power of biology. BBSRC is one of the 9 councils that make up UKRI, which is a public funder of research and innovation that invests to create new knowledge for economic prosperity and societal benefit. This focus is reflected in BBSRC's forward look, with our vision to support knowledge exchange and commercialization activities that enable a vibrant bioscience ecosystem that connects world-class researchers with leading innovators to advance national priorities and access shared opportunities. This recognizes the complexity of how biosolutions are increasingly integrated into wider supply chains and the need for flexible and open mechanisms. This priority is delivered within BBSRC by the Knowledge Exchange and Commercialization team, which has an overall mission to enable a more connected innovation system that delivers for bioscience across the UK. The team manages strategy and policy relating to KEC across research, innovation, infrastructure, and skills. And this includes support for translation, capability, venture, and enterprise. The forward look fully reflects UKRI's vision for research commercialization, which emphasizes the importance of supporting this activity within the research base. That concludes the introductory session. We're now going to hear from researchers who were successful in applying for funding and how the award has enabled them to progress their ideas through to impact.
Hi, I'm Caroline Barelle and I'm delighted to get the opportunity to tell you a little bit about a project that was funded by a follow-on fund back in 2012. And it focused on our Solamer technology, particularly a domain called EO6 and its ability or potential ability to extend the half-life in serum of multiple other therapeutic drugs. So the overarching aim was to use this funding to produce a data-driven evidence package that enabled EO6 to be clinically developed. So looking at the project as a whole, we're very much interested in biologics, particularly antibodies. And what we've developed is a unique antibody-like technology platform. We call these domains Solamers. They're single chain, they're small, they're robust, and importantly, they bind into targets in a very different way from antibodies, often eliciting a new mechanism of action, which is a great differentiation factor when it comes to making a difference in the clinic. This particular project was focused on something we call a biotool. So a biotool is really important. It enables you to do something in the clinic that other domains can't do. And in this case, using EO6, which binds human serum albumin, it can extend the half-life of other drugs for longer. Why does that matter? Well, it matters from the perspective of developing drugs for chronic autoimmune diseases, for example. It decreases the dosing and increases the quality of life for patients who are being subjected to these drugs, often early on in life and for the rest of their lives. So a little bit about the Solamer technology itself. So as I've said, these are single domains. They are in fact shark derived, which is fascinating. These beautiful animals also have other antibodies in their body, but these ones are 10% the size of classical antibodies. And because they're single chain, they have the capacity to be able to be linked together. So really pushing forward the limitations of other antibodies in that they can bind multiple different targets at one point in time, which is really important for complex diseases. And here you can see on the left-hand side, we're developing autoimmune products. These are over an FC region, which is shown in the green, but again producing bispecificity with the red and the blue Solamer domains, targeting different disease molecules. And here again, another level of flexibility is that we're incorporating small molecules to make drug conjugates. But really where these domains stand aside from others is that you can link them together to form bispecific or biparatopic domains and keep the molecule really small. So the final product is a fraction the size of a whole antibody. But as a result of that, it's cleared very rapidly from the body. And so EO6 was designed to bind albumin to be fused to other products, such as our own Solamer domains, to extend the half-life, to enable that therapeutic index, that Cmax, that important efficacy to last longer in the body. And we've used that particularly in our oncology products, as you see here with the radiopharmaceuticals. Endure itself was a success story. We had already isolated it by the time this funding started, but we couldn't show whether or not it, as a shark-derived molecule, would elicit any immune responses, but more importantly, could it extend the half-life of other fused products? And here I'm just exemplifying a few options when it comes to those types of new drug modalities. So we can use it to extend the half-life of our own solomers, as we're doing with our radiopharmaceutical products, but also you can fuse it to single-chain FVs, which are the smallest binding part of antibodies. Down there in the bottom you can see a growth factor fused space Solymer product, again showing that EO6 can extend the half-life of it. And up there in the top right you can see drug conjugates, so again bringing together that small molecule and biologics together, really to create dual functionality but also extension of half-life in the serum as well, so multiple different added on advantages. And EO6, as you see there from the top, we have anointed it as Endure, as it endures in the body. So the project deliverables from the Follow-on Fund were multiple-fold. One, again, these are shark-derived, and if you have any biologic which is not from a human source, you really have to assess it for potential antigenicity. What I mean by that is when it's injected in the body, will it elicit an immune response, which would result in neutralization and potential clearance of that drug. The other big question was, can we fuse EO6 to other molecules, and will EO6 still work? And will those other molecules still work? So that was a critical part of that as well, was looking at the design, the molecular design of these final products. Manufacturability. If you can't make a biologic, you will never get it into the clinic. So looking at that scale up during this project as well. And of course, ultimately, does it do what we want it to do? Can it extend the half-life of other products in the body? So ultimately, this incredible funding enabled us not just to show that EO6 had no immunogenicity, but it enabled us to really bake that into our technology platform, which resulted in learnings for other solomers which are isolated against disease targets. So it expanded our ability to fast-track humanization throughout our platform and product pipeline. It enabled us to do a licensing deal, which is really important for a small company. They licensed Endure to extend the half-life of a particular molecule that they were interested in. And ultimately, while we were under the umbrella of the University of Aberdeen, it enabled us to build an investable, compelling data package, which helped ultimately securing funding for the company to be spun out from the university in 2016. That was when Elasmogen was born. So Endure is now an integral part of our pipeline. As I said before, it's actively being used in our therapeutic products, particularly in our oncology pipeline around the radiopharmaceutical targeting molecules. And the company itself has gone from strength to strength, which really just exemplifies the importance of this type of funding. When you have something which is ready to be translated, it's really important to focus on that and to try and build a company value around it. Lessons learned? Well, as I said, this type of funding is critical when we get to this stage of commercialization, that translatable piece to enable us to do the kind of work that you need to secure investment. But beyond that, I have personally learned an awful lot. I've learned an awful lot about the commercialization of science, what matters, the focus on the end point, so the unmet need. Never forget the patient, because this is a long and winding path, and it takes a long time, and you need the right people around you at those different stages as well. So I cannot do this by myself, and so I would like to finish by thanking the team around me, past and present Elasmogen team, but very, very much UKRI and the Follow-on Fund team, because they played a critical role in the creation and the success of Elasmogen. Thank you.
Hello everybody, I'm Donal O'Sullivan from the University of Reading, and it's a great pleasure to give you this brief overview of a 2-year follow-on fund project just started in September 2024 called FABA+ for short. The project team consists of myself, I'm a legume geneticist, and my academic colleague Dr. Paolo Tosi, who has complementary expertise in seed quality, Dr. Tom Harvey, who's a postdoctoral researcher co-lead, Jake and Rizara, who are technicians, and these 3 folks are all employed full-time by the project to deliver its objectives, and they're assisted by Sarah Allison, who is an intellectual property specialist at the University of Reading. And we're joined on the project by partners, the breeders Sinova and NPZ, and the end user, Atura Proteins, who use large amounts of FABA protein. FABA+ has come about because even though markets for pulses like peas and beans are of course driven by their rich protein content, that protein content, and particularly the specific makeup of seed storage proteins, haven't really featured as breeding targets. And this is understandable because protein composition is a hidden property of the seed, requires destructive and costly analysis to determine, and it's genetically complex, and to top it off, it's negatively correlated with yield. So the nub of our project is that through careful study of both yield and protein content in multiple diverse populations in many sites over many years, we've developed powerful genomic selection models that can be used to break the classical yield protein trade-off. So how would we do this in practice? Well, we follow a somewhat conventional line breeding scheme, whereby we take tens of thousands of unique genotypes from crosses between carefully selected parents, and over 4 subsequent generations, we eliminate all but a few advanced lines, which are both high in protein and high yielding. The process starts with single F2 plants, and we'll use molecular markers in these early generations to eliminate unfavorable alleles. We'll begin the phenotyping in rows at the F3 generation, and there we'll be looking at both yield, potential, and protein content. We'll continue the phenotyping, but now that the number of lines are down to some hundreds, we'll be using genome-wide genotyping to practice genomic selection, and this will enable us to make the final cuts. And the plan is then, at the end, the last few remaining lines will be tested for their processing properties in protein extraction processes. And the best lines will be put forward for plant breeders' rights. Hopefully this diagram here helps to put the specific agenda for the follow-on fund project in the context of the wider breeding process, which is sometimes portrayed as this linear pipeline that can take more than a decade from concept to market launch. And I define the year 0 as the moment when we realized that there was some commercial potential from the insights we'd got through academic research funded by BBSRC, DEFRA, and others, which had enabled us to master high throughput genotyping of the species, obtain markers linked to various quality related traits, and develop these important genomic prediction models that I referred to. Then to kind of cross this important transition, the iCure program, Innovation for Commercialization of University Research, and the Higher Education Innovation Fund were both critical in buying us time and wising us up to some commercial realities that enabled us to propose a follow-on fund project that has now started. And as I said, we hope at the end of these 2 years to obtain these very high merit candidate lines and put them into official variety testing, at which point we may be eligible for plant breeders' rights, be able to sell certified seed and recoup royalties. And it's this last stage of the process. It is not particularly intense because most of the work is done by the statutory testing authorities, but it does require private investment investment. And since you're getting closer to the end point, this is deemed to be the appropriate point where we would look for that private investment. So what would the impact of new high protein varieties be? Consider the simplified view of the current bean supply chain. You have UK commercial bean breeders producing the existing varieties and collecting royalty payment. They supply UK bean growers who on average produce beans, about 600,000 tonnes of beans with a value of roughly 165 million. And those beans are bought by grain traders, grain exporters and animal feed compounders to supply these existing markets: animal feed, aquaculture, and whole dry beans for human consumption in overseas markets. So if Fabaplus breeders were to release these novel varieties, we think this serves new additional markets for in the plant-based protein industry. So there will be new end users, and that will require additional bean growing area, and there'll be a kind of knock-on effect of added value across the whole supply chain. With that, it only remains to thank you on behalf of the Fabaplus team and to invite you to bookmark our website and stay in touch as the work continues.
We'll now continue with information about how to apply and the core scope. FOF is a flagship program for BBSRC and is an integral part of how we invest in research and innovation to advance the frontiers of biology. The Follow-on Fund provides dedicated funding for BBSRC award holders to progress their ideas towards products, services, and processes with the potential to deliver impact. The fund supports early-stage projects that de-risk innovative ideas and later-stage projects with significant technical milestones. Successful applications will be within the remit of BBSRC and will demonstrate scientific and technical merit, economic and societal impact, and a strong potential for added value. They will also have a clear approach to managing any IP. So how do you apply to FOF? Applications are made using TFS and will be accepted until 4 p.m. on the date shown. The project lead is responsible for completing the application process, and this includes making sure it is submitted before the deadline. The application can only be submitted by the lead research organisation. To start your proposal, visit the Funding Finder webpage and click the blue button at the top that says Start Application. This will prompt you to log into TFS or to set up an account, after which you can access the application form. To be accepted for assessment, your proposal will need to meet the requirements of the scheme, and this includes eligibility for BBSRC funding. FOF operates under standard UKRI eligibility rules for Research Council grants. This means you must be employed at an eligible institution and at a suitable level or higher. If you're unsure if you or your institution is eligible, then you can email the eligibility mailbox to check. FOF enables researchers to progress previously supported work towards impact. As a consequence, there is a requirement for proposals to draw upon current or previous BBSRC investments, and to enable research outputs to be further developed into practical application. Eligible previous BBSRC awards include research grants, studentships, fellowships, and work funded by BBSRC through block awards such as IAs, centres, and networks. If you're unclear, you can email the FOF team mailbox to check. Your award will also need to be within the standard scientific remit of BBSRC. BBSRC funds bioscience with a focus upon plants, microbes, animals, humans, and tools and technologies that underpin biological research. This includes work on all scales, from landscapes to molecules. This slide has some common queries about our remit. BBSRC does fund research relating to humans, but does not fund research on specific human diseases or abnormal conditions. In that case, MRC would likely be the relevant funder. To be within BBSRC's remit, the research should be upon the biological mechanisms relevant to the system. For proposals with a focus on technology development, we will often ask if the technologies are platforms rather than clinical or medical interventions. Other areas that commonly lead to questions include animal diseases, where studies primarily focused on animal models of human disease and toxicity are not in BBSRC remit. Research in wild species will tend to be within the remit of NERC, except when they are pests in an agricultural setting or pollinators of agricultural crops, or where they act as a model. For non-terrestrial systems, BBSRC will fund projects related to aquaculture and model species such as zebrafish, but often projects related to marine biology will be supported by NERC. Finally, BBSRC will fund research related to managed land, but only where this relates to agriculture, horticulture, and aquaculture. Project partners are not a requirement for FOF, however they are encouraged as they strengthen most proposals. Partners can be from business or other user groups. Your partners should add value and be connected to the aims of the project. If your project does not have any partners, then we would advise you to clearly explain how users will be engaged in the development and outputs of the work. Partners are required to provide a letter of support to confirm that a collaboration agreement will be put in place if the proposal is successful. Please note that the standard guidance for subcontractors on Research Council grants all applies to follow-on fund awards as well. The follow-on fund is designed to provide flexible options for partnering on awards to enable a clear pathway towards commercialization. Project partners may access intellectual property in a FOF award by making a cash or in-kind contribution. Partners may secure a first right of refusal to negotiate access to foreground IP at the end of a project by making a contribution of 10% of the total project costs. Alternatively, partners seeking to pre-negotiate access to foreground IP must contribute a minimum of 50% of the total project cost. A calculator is available on the Funding Finder webpage for this call to assist you in working through the finances. Empty shell companies with no investment, staff or premises, which are formed for the sole purpose of commercializing foreground IP that is generated during the project, are exempt from contributing project costs. Applicants are advised against the direct assignment of foreground IP to a newly formed spin-out company. Instead, option agreements should be utilized until the spin-out has secured enough funding to drive the translation of the foreground IP, when a substantive agreement should be executed. Your chosen approach for your foreground IP should be clearly justified in the application. Project partners are encouraged to provide clear justification for the value of any cash or in-kind contributions in their letter of support, including how those contributions are relevant to the project. Let's move on from requirements to the assessment of the application. Each proposal will be assessed in terms of its scientific and technical merit, societal impact, economic impact, and added value. The full descriptions of each of these criteria are provided on the Funding Finder page, and this is the same text that we will provide to the assessment panel to brief them on how to approach the assessment. This slide has an overview of the questions you will be required to complete when you are writing your proposal. While I'm not going to read out the text on the web page for each question, the point that I would like to clarify is that you will find a full description of each question on the web page for the opportunity. This guidance includes a description of what the assessors are looking for, and this text is the exact same briefing that we will provide to the assessment panel, so you have complete sight of how we will be asking panel members to approach assessing each question. Please do bear this in mind when writing your responses. These are the core questions in the set that will form the case for support, and I'd like to talk about them in a little bit more detail. The background section is a space to tell the story of how you've progressed to the point where translational funding is timely. Applicants are asked to describe the opportunity and market they are seeking to progress towards to develop or to deploy a new or improved product, process, service, or technology. When describing the route to market, you should consider how the proposed project will progress the innovation towards market and application. The question about intellectual property management and communication is an opportunity to describe how your project will progress the innovation towards translation. This includes your plans to manage IP, any dissemination, and if necessary, the potential to access further investment. In the section about wider benefits, you can describe the impact beyond the immediate commercial opportunity, including the potential societal, environmental, and economic benefits of the proposed approach. Finally, we ask whether there are any ethical considerations associated with the outputs of the project, including those that are beyond formal regulatory and legal frameworks. So I'm going to start to wrap things up now with some general advice for applicants based on how proposals have performed in the past. When writing your proposal, we would advise you to take care to ensure that the proposal demonstrates a clear understanding of what the customer or user needs and how those needs will be addressed appropriately in the FOF project. It's important to focus on the most appropriate opportunity for commercialization and to have a clear development plan. Any risks should be addressed directly with a clear description of any potential impacts and what steps are proposed as mitigation. Management of any intellectual property should be clearly described, including how any foreground IP will be managed. It's also important to have purposeful and clear project partner contributions or user engagement that directly relates to the aims of the project. This is the final slide in this section, and it briefly covers the assessment process. After your proposal is accepted, it will proceed directly to expert panel review, provided that it meets all the requirements. FOF proposals do not undergo external peer review, so you will not receive reviewer comments or be required to submit rebuttals. The Follow-on Fund panel will recommend to BBSRC whether your proposal is suitable for funding based upon the published assessment criteria. And, as always, your proposal should be written for assessment by experts with relevant, but not necessarily specialist expertise. I'd now like to briefly share some other funding opportunities that are available for translational work. The first opportunity to mention is BBSRC iCure, which provides dedicated pre-accelerator support for bioscience researchers to validate their business ideas. This is part of the wider iCure program and is open to research and technical staff at any career stage and at any organisation that is eligible for UKRI funds. BBSRC also supports the UK Innovation, Science and Seed Fund, UKI2S. The Seed Fund provides early-stage venture funding for businesses that emerge from publicly funded research in the UK and has proven itself to be a vehicle for startups to get groundbreaking innovations to market. All of these opportunities and more can be found on the UKRI Funding Finder webpage by filtering specifically for BBSRC. We also offer a dedicated newsletter for opportunities relating to innovation that may be of interest to you. As you're developing your proposals, a good way to locate partners and resources is the Confer platform, supported by the National Centre for Universities and Businesses alongside UKRI. And finally, if you would like to find out about the type of awards that BBSRC and other UKRI councils have made, then all publicly available grants can be found by searching on the Gateway to Research portal. Thank you so much for your attention. I hope that you found the presentation today useful, and thank you again to our guest speakers. We look forward to answering your questions in the Q&A session. Otherwise, feel free to get in touch using the contact details on the screen. Thank you.